The overall goal of this procedure is to establish an acute tissue slice preparation of the mouse vomeronasal organ, or VNO, in order to perform calcium imaging experiments on subpopulations and/or individual neurons in living tissue. This is accomplished by first extracting the VNO from the mouse's nose and then microdissecting it from its cartilaginous capsule under a dissecting microscope in cold ACSF. Next, the isolated VNO is positioned vertically in an agar block, and acute coronal tissue slices are cut using a microtome. To verify the tissue integrity, a few slices are observed after immunohistochemistry under a confocal microscope. Other slices are loaded with a calcium-sensitive dye and then perfused with putative pheromonal ligands, and neuronal activation is observed using calcium imaging.
This method can help resolve key issues in the olfactory field. For example, hundreds of pheromone receptors are expressed in the vomeronasal organ, but very few of them have identified ligands. Pheromone receptors are very difficult to express in a heterologous system. We needed to develop an efficient in vivo assay to assign specific pheromones to specific receptors. We can use tagged mice, where a specific subpopulation of neuron expressed known receptors and can be identified by the expression of GFP. We could also use this assay to pharmacologically investigate ion channels and enzymes involved in pheromone transduction cascade. Demonstrating the procedure today will be Julien Brechbühl and Fabian, Moine, two researchers from my laboratory.